welcome everybody to the first session of the spring series. This is our 11th symposium series, and interesting enough, it's all going to be about water. Before Stan introduces Philip, I'd just like to say that to most life scientists and biologists, we only really consider water to have about three features of it: gas, liquid, and solid, and that it's a rather remarkable solvent for a number of chemicals either outside or inside the body. But it does bathe nearly every process and every biological process we have, and I think it's very much underfeatured in our teaching and our learning. So I'm hoping by the end of this symposium series that we will look at water in a very, very different way. What I'd like to then do is introduce Stan. Now, Professor Stan Botchway has been one of our advisors, well, really from the very, very early days, and he's a pivotal member of our research program. He's a senior scientist at the Central Laser Laboratories at Harwell, and he's going to be giving a very brief talk and introduce our speaker today, Philip Kurian, who is another long-standing advisor of the foundation. So without further ado, Stan, can we open the meeting to you?
Thank you, Geoffrey, for the introduction. And basically what I wanted to do is, since this is the first of the in the series on water, I thought I'll just take the opportunity to basically give a brief and perhaps a basic overview of water. I mean, things that we already know, but perhaps more of a reminder of the things that we knew, but perhaps may have forgotten about water. Okay, so if I can get my slides moving forward. Yes, water. We all have some saying about water. Of course, mine are listed here. Perhaps, water, water everywhere, not a drop to drink. All is water. That's another one of my favorite phrases about water. And then, of course, there is magic on this planet. It is containing water. Perhaps Philip will tell us some about this magic. Water is, of course, as said by D.H. Lawrence, hydrogen 2 parts, oxygen 1, but there is another that makes water water, and nobody knows what it is. All this, I'm hoping, will be revealed in this series. Water, of course, is very special. Obviously, this has been talked about way back in ancient Greece, more than 600 BC. As Geoffrey just mentioned, water is one of the 4 elements, together with fire, earth, and air, was thought to make up what the world is. However, centuries later, with some knowledge, Henry Cavendish discovered hydrogen and reported that it produced water when reacted with oxygen. Lavoisier later reproduced Cavendish's work and gave hydrogen its name from the Greek water former. And of course, this has been reproduced by others, particularly John Ritter, who confirmed water composition by electrolysis using electricity, of course, that we've heard all about. So armed with these basic knowledge in the past of the composition, research began about the strangeness of water. For example, no other material is commonly found as solid, liquid, or gas, or coexisting, possibly in what is known as this triple point on this diagram on the right. As a gas, water is one of the lightest known elements or substances, and as liquid, it's much denser than expected, yet possessing very strong high void space. As solid, it is much lighter than expected as well, compared with this liquid form. It can be extremely slippery and extremely sticky at the same time. I don't know whether any of you have tried this experiment where you've got water between 2 glass planes. It's very difficult to put them apart, and yet these can slide very easily around each other. Water is highly polar and very responsive to electric fields. including those produced by itself. The states can be described by the intermolecular hydrogen bondings necessary for mixing with biomolecules. There are, of course, specific nature of water, the high cohesion between molecules that gives it high freezing and melting points that is essential for life. Large heat capacity, high thermal conductivity, together with high water content in organisms contributes to thermal regulation and prevents local temperature changes and fluctuations. Obviously perfect for body temperature control in organisms. It has unique hydration properties towards important biological macromolecules such as proteins and nucleic acids. Also gives these hydrodynamic properties of molecules, gives it its structure and function. Water ionizes and allows easy proton exchange between molecules, contributing to the richness of the ionic interactions in biology, essential again for life. There is an interesting property of DNA and water, and that is DNA contains what we call 7 water of molecules, and this cannot be removed. It doesn't matter how high temperature you heat DNA at or at what pressures, these 7 water of molecules cannot be removed, which is interesting. We don't know quite why that is. It is, of course, an excellent solvent due to its polarity, high relative permittivity, and small size. So water and biomolecules, very few biomolecules do not mix with water. Obviously, lipids are one of those, or very high polymer substance. Some consider water as biomolecule in itself, as well as a transport medium. It regulates chemical reactions, cellular osmoregulation, temperature, pH, ions, and acts as a buffer. Simply an excellent solvent for life. Aided by small size and multiple hydrogen bondings, clear evidence of water can be involved in proton tunneling in biomolecules. We heard all about this last week in the quantum biology series. There is, however, much controversy about water. One of these being, of course, this idea of perhaps water having memory. The electron distribution in water, which can be seen as rather unusual, where a free pair of electrons, one water molecule, might be able to exert sufficient force on a hydrogen held by a pair of electrons on another water molecule to bind the 2 molecules together. Again, this makes water quite unusual in this regard. The nature of these bondings was much debated by Pauling and Bragg. Much later, Frank and Wen described a new picture of water as consisting of flickering clusters of hydrogen-bonded molecules, and it is these transient bondings that perhaps gives water some of its unusual properties and life-sustaining characteristics. So the water structure, each water molecule is electrically neutral but polar. The hydrogen atoms are partially stripped of electrons and hence partially positive charge, as shown on this top picture, whilst the oxygen atom is partially negative charge. The relatively large positive charge on the oxygen atom nucleus and the closeness of its electrons results in a charge transfer from the hydrogen atoms towards the oxygen atom, leading to a polarity of water molecule. Also, the hydrogen atoms likely possess parallel or anti-parallel nuclear spin, possibly leading to its quantum nature. We also know water molecules can move at astronomical rates through nanochannels. How this is achieved is still not known and under study. There are many anomalies of water that still needs to be discovered. This, of course, being the reactive oxygen species nature when water is ionized, as shown in this equation here. Several of these ROSs can be influenced by magnetic fields, perhaps leading to, again, a quantum characteristics. And again, perhaps some of these might be elucidated by the speakers in this series. So without further ado, I'll introduce our next speaker. So Dr. Philip Kurian is currently a principal investigator and founding director of the Quantum Biology Laboratory at Howard University. His career began as a maths teacher in North Philadelphia before completing a Ph.D. in physics. He is the recipient of several fellowships, grants, and awards, including from the Alfred P. Sloan Foundation, US, Italy, Fulbright Commission, the Guy Foundation, National Science Foundation, and the NIH. His group's pioneering work on single photon superradiance in protein filaments and neuron fibers has been featured by several media outlets, including Science, Quantum Insider, Science News with Sabine, and PBS Spacetime. He was appointed to the chairing committee of the National Academies of Sciences, Engineering, and Medicine workshop on quantum-enabled sensing and imaging for biology. He also serves as a scientific advisor to the Guy Foundation, Science for Seminaries, programs of the AAAS Dialogue on Science, Ethics, and Religion. And without further ado, over to you, Philip.
Thank you so much. I'm glad that I have this great opportunity to introduce and build on what Stanley mentioned. And also just to realize that as we think about water, many of you are coming from a biological and biochemical background. And as I come from a physics background, I'll try to really give no equations at all in this presentation. And so I'll try to give as much of a phenomenological description of water as possible and motivate it from experiments, motivate it from observations, motivate it from cosmology, even cosmological observations that govern the formation of life here on Earth. So this is a bit tongue-in-cheek, but I will try to give the ambition of this talk in the title, and describe how life forming in a very strange and curious substance like water is a unique place and nexus for quantum effects to occur. So, as Professor Botchway mentioned, water is the big elephant in the room, and it is 99% of the molecules in most biological systems, and yet it is probably the least studied in terms of mechanism, pharmacokinetics, pharmacology, and in understanding how the milieu itself in which biology happens guides, directs, coaxes, and intervenes in the processes of life. So I'll pick up where Stan left off, talking about why water is weird. And there's a couple observations, 5 in particular, of why water is weird. So if you notice on the left slide, the anomalously high melting point of water as you go across the RH2 series or down in the periodic table, you can see that water, ammonia, NH3, and hydrogen fluoride, which are all combining highly electronegative elements that essentially grab electrons and hold them more tightly, combined with hydrogen, have these anomalously high melting points and boiling points. And you can imagine, if you linearly extrapolate that line from H2 Te, from H2 selenium to hydrogen sulfide, you would see that the melting point of water should be almost less than -100 degrees Celsius. But anomalously, it has this melting point that occurs at 0 degrees Celsius. And the same for the boiling point. And so this leads us to understand that if you have these forces that are playing a role in water molecules, it must be nearly absent in other members of this RH2 series. So something about having oxygen, fluorine, nitrogen, these electronegative elements bonded with hydrogen produces some very strong force in the medium itself that is nearly absent in other members of this series. So water is actually the liquid we know of with the highest latent heat of vaporization. So this is by far the highest latent heat of vaporization of any known liquid. And you can see again the anomalous behavior of water, hydrogen fluoride, methane, and ammonia. Well, not methane, but ammonia. And this is why these are kind of generally considered as potential solvents for life. But if you look at water, it's very clear that the heat of vaporization is much larger than ammonia at 538 cals per gram compared to 327 for ammonia. A third point of why water is weird, the fact that its ice form, the most common form that we see on Earth, floats because its density is less than that of water. So this is partly because liquid water prefers fewer molecular neighbors, and water ice is even more antisocial. So there's even fewer neighbors because of the hydrogen bonding that we'll discuss. So if you want a visual picture of this on the right, ice here is shown in the upper part of the right figure with its van der Waals radii, and below it's just balls and sticks. But you'll notice those kind of cages that form in the ice. And those cages are about 0.5 nanometer across due to the bond length, right? And so ice in this form, and even in hydrogen bond networks in liquid water, form this sort of liquid crystal-like structure in which processes can occur in a hydrogen bonded network. Now, for comparison, if you look at magnesium fluoride, MgF2, you can see it's a much more closed and compactified space where those caged open rings are not available. So the fourth point, why water is weird. There are 3 phases that exist across the range of our planet's temperatures, right? So liquid, solid, and gas can coexist, or they can exist separately in different temperature locales across Earth. Now, this is of interest because, there are actually 6 different or more types of water ice, but if we have high pressure locales that are shielded from the sun, they need to actually be warmer to sustain life, which actually sets planetary size requirements on our planet for the requirement of a molten core, which can actually warm from below, right? And so that warmth actually just shows these different transitions for the different types of ice, many of which are actually denser than water. But if, for example, water were actually, let's say, less dense than ice, ice would sink to the bottom, freeze even harder, and then over time, all the oceans and all the rivers would be completely solid, right? So the fact that ice floats and that this dynamic equilibrium between solid, liquid, and gas across the planet also gives us room for saying, okay, what are the necessary requirements for life if you have a planetary environment in which the solvent is actually in solid form denser than that in liquid form. And in fact, we would be in trouble, very much trouble, if life in most of the oceans and in rivers were not able to survive during the cold temperatures of the winter. Okay. Now, one thing that's not commonly talked about is that in the transition from the most common form of ice to water, only about 15% of hydrogen bonds are broken, which persists in this considerable liquid crystal structure, even all the way up to the boiling point. So we can really think of this sometimes like a pneumatic liquid crystal as you start to put other objects and ions in the solution. And we'll go to that next. So the fifth point of why water is weird, Stan mentioned this, is extraordinarily high dielectric constant, which is a measure of how much water can shield the electrostatic force between, for example, ionic salts, sodium and chloride, or others. And so you can see here that water next to formamide and hydrogen cyanide is the only pure liquid that has such a high dielectric constant or relative permittivity. Now, why might this be the case? So let's go back to freshman chemistry. We know that there are more or less 4 types of chemical bonds. And I put bonds in quotation marks because, of course, these bonds are ultimately solutions to the many-body Schrodinger equation. And so the many-body Schrodinger equation will give us both eigenvalues and eigenvectors that are then descriptions of the orbitals that define covalent bonds or other structures that we define as hydrogen and van der Waals bonds. But ultimately, all of these emerge from the quantum realm, although we have classical descriptions of them. The ionic bond is the most simple, in which electrons are actually given up, for example, by sodium and given to chlorine to complete their valences, and then they interact through electrostatic interactions. But notice the covalent bond, which is 100 kcals per mole, typical covalent bonds, is about 20 times the value of such a hydrogen bond, which shows up in ammonia, in water, and in hydrogen fluoride, as I showed on the previous slides. But these bonds, of course, are even an order of magnitude stronger than the van der Waals bond, which occurs due to London dispersion or similar dispersion forces. And so for the biologists, a comparison is that ATP hydrolysis is only 2.3 kcals per mole stronger than a hydrogen bond. So the fact that we have these hydrogen bonds is something that we need to take into consideration when we're thinking about the chemical efficacy of biological reactions and reagents. So the elephant is in the room, and we are going to talk about how this elephant plays a role in the organization of life. So here's some hydrogen fluoride bond networks, because it's not only water that forms these hydrogen bond networks, but you can see from this picture that I've taken from John Barrow's and Frank Tipler's book of the anthropic cosmological principle, you can see that these hydrogen bonds can form these quasi-periodic lattices. They can form certain regimes, but that are linear, but they can also form these similar proto-micelles. And could they be places for organizing life? Well, hydrogen fluoride is a bit problematic, but because it doesn't form these tetrahedrons that you see in the water hydrogen bond networks. And so we know that methane has the ideal tetrahedral angle at about 109.5 degrees. But because of the lone pairs of electrons that complete the valence of oxygen, you kind of smush that down so that you have water's quasi-tetrahedral angle is about 104.5 degrees. And you can see the others are distorted from that. And the variation in that bond length, the fact that it's not a perfect or ideal angle, leads us to believe that there may be some approximate but not exact symmetries that may allow for different types of structures and the diversity of structures that we see in inanimate as well as animate objects. So, of course, these hydrogen bond networks end up looking with more space, as I showed you in those hexagonal lattices, with these kinds of spacings that are shown in angstroms and with that 104.5 degree bond angle. And of course, with these networks, as I showed, you can have nonpolar solute molecules that can then dissolve or be hydrated in these cages in which water molecules form around them. Okay, so how does life form in this aqueous environment? This is a nice little article that was written by George Wald in the 50s in Scientific American. And he asked the question, and took very seriously how did living matter first arrive on the Earth. And Wald was a biologist, but he ended up learning a lot of physics in the process. And he starts this article by actually going to the account, the biblical account in Genesis, where it's certainly not chronological if you're a student of the Hebrew, but it is actually quite interesting in the way that creation is formed in that biblical account. And so from that, the idea of spontaneous generation of life was something that was long considered for many, many centuries by scientists, scientific thinkers, and theological thinkers alike. But the Wald wanted a mechanism, wanted a way to understand how could the waters, let's say, give rise to living organisms and creation. So a key argument that is not often appreciated, but it's rather simple, just takes, even an elementary school student can do this, is to think about rare events, extremely rare events, and to think about their probability if given enough trials over time. So if you have an extremely improbable event, let's say for the sake of argument, 1 in a million, okay? The chance the improbable will occur is, of course, a power of that million to the -1, right? So if I do this many, many, many times, I'll get very, very close to 0 in terms of the probability of that event, which makes it highly improbable. But now let's say to get life going, I just need one critical event to happen. I need some critical event to get over that hump. And if I get over that hump, I don't have to do it again and again and again. And so let's think about the chance that an improbable event will not occur. So if it's 1 in a million, then it's 999,999 over a million that improbable event will not occur. And now if you take this number, of course, that's very close to 1, and you raise that to the x power, and you give x sufficient number of trials, let's say 10 to the 5th times, okay? Then we have a 10% chance, 1 minus 0.90, we have a 10% chance that that improbable event will occur at least once. And if we raise that to 10 to the 8th, which if let's say the event occurs once per year, you have 100 million years, which is certainly in Earth time of 4.5 billion years. This makes it 10 to the -44th, and 1 minus, that's essentially 0. And so it's a near certainty that an improbable event will occur at least once. And this is kind of the argument for how improbable events can become highly probable given cosmic times. So people who wanted to explore the experiments and the phenomenology began, I think many of you will be familiar with the Urey-Miller experiments, where you have kind of an electric discharge to simulate lightning in a highly reducing atmosphere. And then the formation of amino acids or their precursors, called Strecker precursors in the chemical cycle, form. So that's one experiment that people are very fond of citing in the early origins of life experiments post this article by Wald. There's also the formation of sugars from atmospheric formaldehyde by Butlerow. And then also there's Oro's experiment showing adenine and other heterocycles can form from polymerization of ammonium cyanide in solution. Now, the problem with these reactions, even though they're one pot, you can mix things together and you can get them in one single kind of chemical reaction chamber, but they are multi-step. So they suffer from really low overall yields of biologically relevant products because of unfavorable reaction sequences that are either side or downstream from the ideal biologically relevant product. Okay. So in 2015, John Sutherland's group actually published this work in Nature Chemistry, showing that you could actually, by sequential delivery of certain reagents and with a dash of UV in this hydrocyanide soup, you could actually produce all of these different biologically relevant products, lipids, amino acids, nucleosides, as well as some sugars, okay? And so if you look at a lot of the astrobiology research and the origins of life research that go out into space, many of them who look at asteroids and who look at different icy bodies in our solar system are looking for planetary brines, right, that contain these kinds of salts as well as ammonia, which can drastically depress water's freezing and elevate its boiling points, right? So why is this important? Well, in other celestial bodies, like Ceres, the dwarf planet between Mars and Jupiter, or out in near the Kuiper belt with Jupiter's moon Europa or Saturn's moon Enceladus, you've got these environments where the temperatures are well below water's conventional freezing point, closer to the sun here on Earth, right? So we have these opportunities for extremophilic life only if we have sufficient amounts of these kinds of primordial salts that could, in a certain environment, produce life, maybe not the kind we're accustomed to, but similar forms of life, maybe carbon-based, most likely, that form these chains that can then produce polymers that can then process information common to the ways we're accustomed to with life. And in fact, several space agencies have taken this very seriously. Ceres was visited in 2015 by NASA's Dawn mission. Jupiter's moon Europa was there was a Europa Clipper mission that was launched just last year, and I think it just did a gravity assist around Mars. And then there is a mission plan to Saturn's moon Enceladus in the late 2030s that will probably arrive there in the late 2040s and explore Enceladus in the early 2050s. So that early kind of multi-step, multi-reagent mixture in the primordial soup was this Sutherland Group article that they call a cyanosulfidic protometabolism. So all these precursors could form in a reaction system where there are key cyano carbohydrates as well as sulfido carbohydrates, okay? And so there's something called that I was unaware of before I prepared this lecture called the Kiliani-Fischer chemistry. And this Kiliani-Fischer homologation chemistry, it requires a sequential and periodic delivery of hydrogen cyanide and hydrogen sulfide with other reagents. And this raises the very interesting question that I'm sure Alistair and others have thought much more about, is about how this kind of chemistry could be produced in these prebiotic or early Earth hydrothermal vents, right? Which we know these black smokers and white smokers at the bottom of the ocean in the Mariana Trench and other places, okay? So I thought I'd talk a little bit about supercriticality. Stan mentioned it in his introduction, talking about how either nano-confined water, it's been observed, or water that's confined to tubes has an extremely high flow rate, extremely high predicted permittivities. So this is in confined spaces. But supercritical fluids in general, the distinction between liquid and gas becomes much more blurred, and they are superior to gases, though, in dissolving the materials for life. So, for example, if you have these gas giant planets where they're mostly made of helium or hydrogen, or let's say methane and ethane, as in a gaseous form, in supercriticality, some of these become much better at dissolving the materials for life, and water is no different. So fine-tuning near that critical point produces very large changes, not only in density and viscosity, but relative permittivity, which allows, of course, the dissolution of ionic compounds and the delivery of those ionic compounds elsewhere. So if you take ionic minerals from, let's say, hydrothermal vents or from another source, and you have water with an extremely high dielectric or a high relative permittivity, you can then transport those materials to a lower pressure or lower permittivity, depositing or precipitating these compounds nearer the surface, which can then allow UV photochemistry to take place. As I mentioned, it's been observed and predicted by molecular dynamics that nano-confined water in cylindrical nanotubes has about 10 times the bulk value. So it can certainly dissociate ionic compounds much more strongly if this is true. And these are ongoing debates in the quantum chemistry community about how, if you take a dielectric constant estimate at extremely high temperature and pressure for water, this depends critically on the quality of what's called the exchange correlation functional that's used in density functional theory. And for those of you who don't know, density functional theory is a way of reducing a very complex problem, which is the many-body Schrodinger equation, for all the electrons in your system, which for water is even non-trivial for a single water molecule. But reducing that extremely non-trivial, very difficult to solve and impossible to solve in one go. It must be solved in a self-consistent manner iteratively. How do you do that? And density functional theory is one method to reduce the degrees of freedom to a vector-valued scalar function over the three-dimensional space of your molecule. So at each point, you have a density rather than tracking the quantum objects, okay? But there's an active debate about how do you measure these things in a quantum chemistry or even predict them in a quantum chemistry or electronic structure framework. So another important thing to remember is that the creation and usage of carbon dioxide, both as a waste product from respiration, but then as dissolved in a supercritical fluid or as a supercritical fluid that is nonpolar, can serve as an effective hydrophobic solvent for facilitating dehydration reactions in water. So if you have a nonpolar kind of oily mixture that can separate from water and then produce a space where you can have dehydration reactions, you then have a mechanism for producing things like the lipid bilayer and other types of nonpolar side group reactions that can then generate in an aqueous medium the constituents of organized life. So here's a visual representation of that. I won't go into all the details of these different types of hydrogen bond configurations at the interface of polymers, but in the next lecture, in lecture 2, my colleague Ali Hassanali will probably talk a lot more about how both nuclear quantum effects and electronic quantum effects, and I'm not talking about the cell nuclei, I'm talking about the atomic nuclei, of course, but these tunneling effects, as well as these electronic density configuration changes across many water molecules in the hydrogen bond network, can drastically change predicted properties of interfacial water relative to bulk water. So since the early 2000s, it has been known through terahertz vibrational spectroscopy that there is an intimate dance of water surrounding proteins. And this can be several nanometers from the surface of such proteins. And this is just one example from Martina Havenit's terahertz spectroscopy group showing the strong correlation between these water networks around proteins that extend extremely far from the protein surface, much further than before. And in fact, if you have these water molecules that are surrounding these biomolecular polymers and you have a situation where the dipole moment, the permanent dipole moment, which is large of most proteins, can be extended even further. And so imagine you no longer have a protein that's oscillating back and forth in a thermal environment or in a non-equilibrium thermal environment, but now you have an extended and enlarged dipole, essentially acting like an antenna in solution that can transmit electrodynamic information classically, just as a cell phone would in a in our context. Okay. So it's important to remember that the electrodynamic milieu is the organizing framework for speeding up the process of architectronics in the cell. So here's an example of collagen filaments in a very dilute acetic acid solution. And then when you add 1% sodium chloride, look at the beautiful polymers that form. And now this is very interesting because if you increase that concentration of ions in solution, you're actually dampening and screening electrostatic effects. So take a look again. You have collagen filaments in a very dilute acetic acid solution. You add sodium chloride, and then you get these beautiful polymers of collagen. Now, if electrostatics is your answer, you'd be wrong because the sodium chloride in the solution actually exponentially screens out those interactions beyond just a few nanometers. So it was known actually at the time, even at the time of Maxwell and possibly before, that long-range interactions occur in the aqueous environment at above oscillating fields above about 250 megahertz. So above 250 megahertz, these are transmitted due to the dielectric permittivity, which is frequency dependent in water. These are transmitted at long range in the cell. The dielectric permittivity at petahertz of water is about 4, whereas at the standard dielectric is about 81. So you can see the change in that dielectric has enormous consequences. And as these polymers form, of course, they come out of solution, but they can also, because of their hydrophilic side chains, they can form interfaces and surface layers with water, which then allows other types of chemistry to form proximal to the aqueous environment. And this is sort of a proto-life system that can form a priori before animate life begins. Okay, so I'm going to dive a little bit deeper into the quantum realm. So for the sake of time, I will refer you to a recent paper that was accepted in Science Advances of mine that will be published later this month. So I encourage you to take a look that fleshes out this argument much more deeply. But at least intuitively, let's take a word from Milton, cycle and epicycle, orb and orb. And we are all beholden to cycles in the cosmos, as well as cycles at the human scale, and cycles that are much faster than our conventional human experience. And so what do the hierarchy of those cycles look like? Well, quantum mechanics tells us that the smallest scale at which those cycles can occur is the Planck scale. And ultimately, that's governed more or less by Planck's constant, which the Planck time, if you want to reference, is 10 to the minus 43 of a second. So here we have the Planck length scale, which is about 10 to the minus 33 to 35 of a meter. But of course, living systems operate in a span that we are comfortable with saying of 12 to 18 orders of magnitude that are far from the Planck scale, at least in our conventional perception. So we might be comfortable saying, okay, well, I'm comfortable with saying things occur at the picosecond scale for thermal motions, and then of course for enzymes at the microsecond scale and the millisecond scale, and then of course with human beings at the seconds and hour and year scale. And so we have living systems that operate in the middle of this hierarchy. And how do we connect that to even cosmological scales at the level of the Hubble constant, that we measure the size of the universe at 13.8 billion years, which you can convert to a size estimate. So I want to highlight some recent work from my group, as well as with coworkers and collaborators. So in 2023, we published this work in PNAS Nexus with Ali Hassanali that was an atomistic molecular dynamics simulation study looking at how if you photoexcited particular chromophores of tryptophan in a bovine serum albumin protein, how you can generate large-scale vibrational collective modes. So these are 3 orders of magnitude difference in scale in terms of the electronic excitation time and the fluctuations that occur due to vibrational degrees of freedom according to the nuclei of the protein atoms, right? But what we showed was that by exciting these tryptophans in the BSA, you can actually produce a condensate of modes in lower vibrational modes, much slower modes. So what was commonly associated with the femtosecond scale then becomes something that occurs on the nanosecond to microsecond scale, which we were able to track the vibrational breathing modes of these proteins over up to nearly half a microsecond. And so this kind of idea that is not new. Herbert Froelich in the 1960s had the idea of pumping things electromagnetically and then seeing these kind of breathing modes in the cell or in biology in this way. But actually with Giuseppe and with others, in 2018, we published this much more theoretical physics-based work to look at how, for example, no one disputes on the left this Feynman diagrammatic picture between 2 electrons. So 2 electrons, they interact through a photon represented here by the variable gamma, because the photon is the quantum of the electromagnetic field, okay? So no one disputes that. But in an aqueous environment, and this is work that Giuseppe and Emilio Del Giudice and Preparata and others developed in the 80s. They were coming from this Italian school of particle physics that then emerged and wanted to try to bring quantum field theory into understanding the electrodynamics of aqueous dipoles, right? And so if you imagine this milieu of water dipoles that is proximal to proteins, DNA, other objects, it's even been observed that there is a hydration shell, of course, not only around proteins, but as Stan mentioned, around DNA. And so what we wanted to do was to explain this even at the level of a single molecule of DNA or any sort of electromagnetic impurity in the aqueous environment. And essentially what we showed using the rotational, quantized rotational degrees of freedom of the water molecule is that by putting this electromagnetic seed in the aqueous environment, you could stimulate long-range polarization behavior in the aqueous environment. And yes, so that was a lovely result that has been confirmed in some other experiments by Marco Bettini's group, shown here in the PRX 2018, which is what then motivated our 2023 molecular dynamics work. So a very simple observation, if you didn't follow what I said previously about quantum field theory. If you put a box of water in a UV environment, okay, the wavelength at UV scales is approximately 300 to 400 nanometers. Of course, it can be a little less or a little more. But if you have an architecture of cold water molecules, right, the spacing due to hydrogen bonding is on the order of about half a nanometer. So in a volume of lambda cubed, if I take 300 nanometers cubed and I divide it by half a nanometer cubed, I get about 100 million water molecules, right? So remember, the next time you see spectra of water, liquid water, water with stuff in it, these different solutions that you might see, remember that these spectra are collective signatures. They are not signatures of individual water molecules, but rather they are signatures of a collectively coupled medium. And that collectively coupled medium has different quantum effects than a single water molecule. And if you take nothing else from my talk, that's a very important thing, because many researchers, not my group, but many other researchers have spent years thinking about how do these collective effects actually have significant influences on what's going on in the aqueous environment. Okay, so in the last few minutes of my talk, I want to connect to the cosmological scale. And so there's this beautiful article in Physics Today from 2018 that described something called an astropause, which is a polycyclic aromatic hydrocarbon. And so these polycyclic aromatic hydrocarbons, or astropause, astronomical polycyclic aromatic hydrocarbons, these actually form around in circumstellar clouds, so around stars, and they actually also form in the interstellar media. And they're essentially flakes, they're essentially planar flakes of fused benzene rings. But interestingly enough, in the interstellar media, where there's a UV-rich environment, there's no absence of UV photons, for example, around stars, you can actually amputate hydrogens, and you can even have these sort of breakage of carbon-carbon bonds in the benzene ring. And when that happens, instead of just having the hexagon of benzene, you can actually form a pyramid of, a pentagon of benzene, a pentagon of carbons next to a hexagon. And for those of you who know your organic chemistry, a hexagon and a pentagon fused together is an indole ring, which is the basis for tryptophan, for serotonin, for kynurenine, for many of these signaling molecules that we see in biology. And it's been known that there's a lively debate going on right now in the astronomical community about whether there are interstellar signatures of tryptophan in the ISM. And so there's been a debate the last 2 years about whether it's really a signature of tryptophan. So we await those results eagerly. Why? Well, the elemental composition of life is based not only on the elements, the carbon, nitrogen, hydrogen, oxygen, phosphorus, and sulfur that we're commonly aware of, but also on these aromatic heterocycles. And these aromatic heterocycles form our DNA bases like cytosine, uracil, and thymine, but also adenosine triphosphate, which has a very similar indole type ring as tryptophan. You might be asking now, why not silicon-based life over carbon-based life? Well, one of the problems with silicon is that when silicon polymerizes, because of its pi orbital differences to oxygen, it actually forms quartz rather than the plethora of diverse polymers that carbon can form. And there are other energetic reasons for preferring carbon to silicon-based life. However, we do know that silicon carbide polymers do form superradiant states, and they do form in the interstellar media. So there's opportunities for other forms of life outside of carbon-based systems. So how does water figure into our picture of the early universe formation, as well as early solar system formation, and then early Earth formation? So if we go back 13.8 billion years to the Bang, and then we go advance 10 billion years for the time that's required for carbon to form in stellar furnaces. And then we have asteroids and meteors that can then deliver, let's say, tryptophan and other biological precursors to Earth. we then have a mixture of things coming exogenously from Earth, but potentially also delivered from below, from hydrothermal vents and other places in deep sea environments that don't have access to UV radiation. So we have a marrying of cosmic UV and chemosynthesis that then meets again on Earth in this rich UV photochemistry, thus generating the emergence of life. So respiration, we all know, is a very symmetric equation in the following sense of chemistry, right? You produce, for a molecule of glucose, you have 6 of oxygen, molecular oxygen, and you produce out 6 of water and 6 of carbon dioxide. And so these products, because carbon dioxide can be dissolved and it's easily a gas at common room temperatures, these are easily disposed of in any environmental form. This is not the case for ammonia chemistry, which would probably happen if you have an ammonia ocean, right? And these are liquid products or solid products that are not easily gaseous at the temperatures of outer space. One other observation. We recently had this landmark publication showing single photon superradiance in cytoskeletal filaments. Now, one observation that's been made about photosynthesis is that chlorophyll is a terrible absorber of our natural sunlight wavelengths. And the question is, why is it that chlorophyll would be so unoptimized for the absorption spectrum of the sun? Well, one idea or hypothesis is that perhaps a weakly absorbing environment in the visible spectrum, not in the UV now, but a weakly absorbing environment is precisely the type of Goldilocks zone in which you could excite these field-directed behaviors that allow for self-organization, but at the same time do not overpower these aromatic networks and break bonds like you would find in the interstellar media. So I leave you with this curious little poem, just to remember how funny and weird and beautiful water is. So thank you so much for your time, and I look forward to your questions.